their colleagues, their academic community member of the Babeş-Bolyai University. Welcome to a new presentation of the conference series organized within the Studium Generale program of the Star UBB Institute, the interdisciplinary meeting space of teachers, researchers, and students of the Babeş-Bolyai University. This week, the brain will be in focus. We will take part in a presentation related to neuroscience, with title Hunting for Brain Oscillations, Functional Role and Estimation Techniques. Our guest today is Raul Mureșan, a senior researcher at the Star UBB Institute and president of the Transylvanian Institute of Neuroscience, Cluj-Napoca. He is a principal investigator at the Experimental and Theoretical Neuroscience Lab of Teens. He is from Cluj. He was bachelor student at the Technical University of Cluj. Then he obtained his PhD degree from the Technical University of Cluj-Napoca in collaboration with the Neuroscience Department of Frankfurt Institute for Advanced Studies and the Neurophysiology Department of Max Planck Institute for Brain Research, Frankfurt am Main. Then, between 2004 and 2007, he was a researcher at the Frankfurt Institute for Advanced Studies, where he was working in the field of theoretical and computational neuroscience. His research interests are related to neurophysiology of the neocortex, emergence mechanisms and functional role of neuronal oscillations, artificial neural networks and simulation, models of intelligence and self-organization, and all the topics related to neuroscience. He has a wide scientific activity with many active scientific collaborations. Before inviting Raul Mureșan to introduce us to this very fascinating topic, I would like to remind the audience from the internet that they can also ask live questions on the Babeş-Bolyai University's YouTube channel, where the conference is broadcast. Raul, thank you for accepting our invitation to be here at the Taylor Bastion of Cluj, and we look forward for your presentation.
Thank you very much. It's an honor to be part of this presentation series. Thank you for inviting me and for introducing me. So today we are going to talk about brain oscillations. Brain oscillations are some processes that are all over our brain. Even as I speak, I'm using brain oscillations to produce other oscillations, which is speech, for example. So as you will see, brain oscillations are everywhere, and most likely they organize the way we think. So first we'll start with a little bit of a background, and I will show you what we do in the lab. The questions that we ask mainly relate to mechanisms of perception. For example, we want to find out how we see, how we perceive objects in the real world. The second kind of question relates to the function of neural oscillations. Although you probably heard about various oscillations like alpha, delta, beta, gamma, there is still a wide scientific debate if they really carry some functional role in the brain or if they're simply an epiphenomenon and they correlate to normal brain activity. So this is still an open scientific question at scientific level, and we're still working hard to find out what's going on. Then we are also interested to understand how the properties of individual cells, neurons in the brain, shape the activity of neural circuits. This is also something that is very, very complex, and we are at the very, very beginning of this field. Finally, we want to understand what happens in the brain at brain-wide level, to understand how these different areas communicate, how larger circuits give rise to perception, support cognition, and behavior. So all these are fields that actually are investigated in our lab. And among the techniques that we use, the first one, which is the maybe easiest to apply but the hardest to understand and analyze, is EEG in humans, electroencephalography, which means recording the electrical activity of the brain. This is done with amplifiers, with sensors that are put on the scalp, and you can pick up the very, very faint signal of the brain, and then you have a really hard time analyzing it. So if recording the data takes 1 hour, analyzing and understanding what's going on can take years. The second technique we apply is called in vivo electrophysiology, and this one is done by implanting electrodes directly into the brain. This can only be done in animals for ethical reasons, and we use various models of animals like mice. One can use rats and mainly lab animals. The advantage of this technique is that you can look in the very circuit of the brain. You can hear single neurons firing, and you can see how local circuits are organized. The disadvantage is that you see a very, very tiny piece of the brain. So if with the EEG you see the entire brain, but from the satellite, with the microelectrodes, you see locally the grass, the local grass in the field, and very tiny pieces of cortex. Finally, another technique that we teach at the summer school that we organize and we plan to apply in the future in my lab is using lasers to image brain activity. This is called optical imaging. And simply put, what you do is you either genetically modify the animal or you use some viral vectors to infect cells such that when they fire, they light up, they become fluorescent. And using this kind of microscopy, you can then see individual cells lighting up like stars in the sky, right? The advantage of this technique is that you can see thousands of cells at the same time. The disadvantage is that this relies mostly on calcium signals, which are still slow. Okay, so what we do in the lab is not neurology, it's neuroscience. And people are often confused what neuroscience is. It is very important to stress out that neuroscience is not a single field of science. It's probably one of the most multidisciplinary fields in research, because it includes biology, psychology, medicine, mathematics, physics, people who do chemistry, computer science modeling. Even the field of neuroeconomics exists, or social sciences are also more recently driven and inspired by what we find in neuroscience. So neuroscience is somehow an aggregation, a melting pot of multiple scientific disciplines. Okay, so this is the background of what we do in the lab, and now we're going to start talk directly about oscillations. So what are brain oscillations? If you record the activity from the brain, this is a trace that was recorded in the monkey visual cortex in Frankfurt, at the Max Planck Institute for Brain Research in Wolf-Zinger's lab. And what you see there, the blue line, is actually a local field potential, which means an electric potential recorded in the brain. You see with the red dot, I actually outlined the slower rhythmic oscillation. So basically a brain oscillation is a rhythmic modulation of activity. It's a wave, if you want. What you see here is that you have a slower wave, and on top of the slower wave, I can actually use the pointer. Can you see? Yeah. So on top of the slower wave, you have these faster oscillations, right? So you already see that there are two rhythms that coexist. Now, why are these oscillations important? There are different theories. We actually don't know. There is no single theory that has been 100% proven. This is all speculation and all hypothesis, and still a very active field of investigation. So first of all, what we know is that oscillations correlate with perception. This is a famous, on the left, you see a famous experiment performed by Wolf-Zinger in the late 80s, where they recorded different cells in visual cortex, and they used either a single bar that would excite the receptive field of these cells, or two different conflicting bars. That's how they are called. What you see is that in the first case, when there is a coherently moving bar on the top left, A and B, the two cells start to fire in synchrony, and not only in synchrony, but they start to oscillate together. Whereas when you use locally the same stimulus, because locally in the receptive field you have the same local bar, but they are not connected, then you see no synchronization and no oscillations. So this led to the hypothesis that such oscillations actually contribute to dynamical binding. And one such example is on the right. This is a typical example of a multi-stable figure. You can either see a vase, you can see a single face, or you can see two faces from the profile, right? And the question is, how does the brain change perception to the same image falling into the retina can generate multiple different perceptions. How? One possibility is that neural oscillations and synchronization of different assemblies of cells in the visual cortex actually binds the local visual features in one way or another dynamically, depending on the context and on the actual perception that you generate. So with the same image on the retina, you can generate multiple different perceptions. And one idea is that oscillations contribute to this. Another idea is that oscillations couple different areas in the brain. You have to know that the brain, at least the human brain, is hugely, massively connected. Between almost every 2 single cells in the brain, you have on average at most 2 synapses. It's almost like everybody talks to everybody else. So how do you prevent the brain from becoming epileptic if everyone talks to everybody else? One idea is that oscillations mediate who's talking to whom. And one example is on the right. We have 3 neurons. We have the red, green, and black. And you see that 2 of the neurons have oscillations that are synchronized. When one is up, the other one is up as well. Whereas the black one is in anti-synchron or anti-phase. When the other 2 are up, he is down. So this means that neuron 1 and 2 will be able to exchange spikes and messages because they're both excited at the same time. Whereas with the third neuron, they will not communicate because when they talk, the other one is inhibited. So on the same connections in the brain, you can multiplex who's talking to who by simply dynamically organizing brain rhythms. And this has been proposed by Pascal Fries. It's called the communication by coherence hypothesis. So this is what we know about oscillations. Of course, there are also the slower oscillations that we are going to talk about, which are expressed in sleep and which are probably helping the brain renormalize synaptic activity such that it can learn again the next day. But we're going to talk about that in a minute. So first of all, how do oscillations emerge in the brain? It turns out that even the activity of individual neurons is prone to generating oscillations. If you have a single cell, and this is depicted here on the left, and you inject a current into a single neuron, it will start to fire periodically. And then there's this kind of discharge and the charge phase. This is called the relaxation oscillator, and the frequency depends on how strongly you drive the cell. The more you drive it, the faster it will fire, the higher frequency, right? So even the single neurons in the brain are actually relaxation oscillators. Second class of mechanisms is the so-called push-pull mechanism, where you have 2 opposing forces. You have excitation and inhibition. And what happens is that excitation builds up and then fires the cells, and then these cells make the inhibitory cells fire, which actually produce inhibition, which bring the system down again. And this whole process repeats when inhibition is extinguished. This is the typical mechanism in the pyramidal interneuron gamma mechanism, which is a fast oscillation. Also, in the same class, you can generate oscillations if you have simply inhibitory networks and you have some kind of tonic drive that makes the cells fire, produce inhibition, bring them down again. When inhibition goes away, the system revamps. This is called interneuron gamma. But both of them are class of a push-pull mechanism. So you have some excitation and some inhibition, and the 2 forces fight each other, right? Another class of mechanisms that is actually very non-trivial, and this most likely contributes to the oscillations that happen in your brain when you sleep. The brain is highly recurrently connected, which means that if you throw a match, it will blow up. It will blow up, but then inhibition comes in and brings it back down. And what happens, it just turns so that if the circuit is it's called a quasicritical state, in the sense that it is stable, but a single tiny pinch will make it explode. This creates a so-called quasi-periodic network bursting. So what you see here is from a paper that we wrote in 2007, where we have a circuit that has random inputs. And because it's strongly recurrently connected, at some points you see that the activity doesn't blow up. It has tiny fluctuations, but from time to time, by chance, this fluctuation is self-amplified by the feedback loops, and then you have a strong network burst. This is a mechanism that produces slow oscillations, but the same mechanism is very likely the one that is behind epileptic seizures. And this is why it's very important to understand such circuit mechanisms and to realize how to dismantle them. For example, you can actively inject currents such that when such a burst is about to emerge, you can bring it back down. Peter Tass is doing this actively in epileptic patients in Germany. Yeah, so yet another mechanism relates to the properties of individual cells. Most of the cells in the brain, the big pyramidal neurons, act as integrators. The more you drive them, the more they fire. But there are some tiny cells which exhibit so-called membrane resonance. They will only fire, or preferentially fire, if you drive them with a proper frequency. This is called a bandpass filter or resonating filter. So the cells will resonate to some kind of frequency. Now, we have studied this actively, and this is something that is very, very little studied so far in the brain. We really don't understand what resonance is doing in neurons. We're just now beginning to explore this. But what is important and what we found out in the past years is that resonance itself is not sufficient to produce oscillations. It favors them, but it doesn't produce them. What you need is a circuit. So in this particular example, we did experiments with cells, excitatory cells, which act as integrators, and inhibitory cells, which act as resonators. And what happens is that if you have only integration, so excitatory integrator and inhibitory integrator, then what happens is that you can generate oscillations, but they are not stable in frequency. On the left side here, you see that if you have a varying input, then you will generate these nice oscillations that you see, but the frequency will also run away. If you replace the inhibitory cells with resonators, then suddenly everything stabilizes. You get very beautiful oscillations, but also very stable in frequency. So just by playing with the properties, the memory properties of different classes of cells, you can actually change the properties of oscillations, their expression, and how they react to input, how they're generated, right? However, the mechanism by which resonance stabilizes oscillations is very complicated. And it could probably be modeled mathematically, and it requires some heavy mathematics. I will also talk about the work of Pikovsky, one famous mathematician who's working on synchronization of weakly coupled oscillators. But going back to the subject, why does resonance stabilize oscillations? So it took us 3 years to figure this out. And it turns out that what happens is that you have excitatory cells fire, here in blue, and then excitatory cells produce a wave of excitation that excites the inhibitory cells, which come and fire a bit later. What happens if you don't have resonance in the circuit is that the more you fire, the stronger the input, the faster will the inhibitory cells fire after the excitatory cells. And this can create a so-called inhibitory-excitatory volley interference, such that inhibition overruns excitation and destroys the cycle. And when you destroy the cycle, suddenly inhibition is dropped down because not so many excitatory and inhibitory cells follow. You reduce the inhibition and your frequency runs away. What happens with the resonators is that it prevents this interference. Because resonators like their own pace, they stay away from excitation, which means the more excitation you produce, the more inhibition will follow, and they will balance each other out, and you will get a beautiful clockwise oscillation. Now, what is important is that it is known in the brain that single cells can change their properties. They can be integrators or resonators, and by neuromodulation voltage, they can shift from one regime to the other. It could be that when you are sleepy, you're more like in an integrator regime, whereas when you're really aroused, you have beautiful, strong clockwise oscillations, and this is something that is known in EEG, and maybe your resonance in your interneurons contributes to this. Another mechanism that helps stabilize oscillations is synchronization. So this is a bit of a complicated diagram, but just think about the fact that you can have a cell firing anywhere here. And then you apply a strong inhibitory pulse. The stronger the pulse is, in the next cycle, you bring everything together. So what happens is that if you have resonance, inhibition automatically resynchronizes your circuit. If your cells were firing randomly and you get a strong inhibitory pulse, this realigns them and makes such that the oscillation comes back and becomes much more precise because the populations will align to the oscillation again. Finally, This is a really complicated plot. What is really interesting is that the oscillation frequency in such circuits is not the resonant frequency of the interneurons. It's always higher. And this is because resonance is a dynamical process. And you can, here what you see is excitation and inhibition as a function of the state of the cell, where it is in the firing cycle. So depending where you hit it, you can accelerate or slow it down. And although your resonance frequency is at 40 Hertz, you will engage in oscillations at 50 Hertz. So the way things happen here, the dynamical interaction between current, the state of the cell, produce oscillations of different frequencies and different properties. And this is very, very complicated to understand, even in simple models that are simple differential equations, coupled differential equations, let alone in the brain, where we didn't even yet measure these things properly. Okay, so what we have seen is that producing oscillations can be done through various mechanisms, and some of these mechanisms are very far from trivial. They're very, very complex and complicated. Now, what are these oscillations good for, and how are they classified? So, anecdotally, they have been discovered by Hans Berger, who was in the army during First World War, and the story goes that he fell from the horse, and his brother dreamed of the fact that he would break his leg, but he couldn't have found out in time. So there was this idea that somehow they communicated telepathically. And he started after the war to study the brain by using simple galvanometers. And what you see is actually a trace that he recorded. And the first waves that he discovered are the so-called alpha waves. So if you close your eyes, in the occipital cortex, you will see very strong alpha waves, which are between 10 and 12 Hertz. And they are so strong that you can measure them with even primitive instruments. Later, he discovered the so-called beta oscillations, which are a bit faster. And almost everything that has been done in about 70 years was done mostly empirically. So people have discovered the alpha, the beta, then the delta, the gamma, and these are all different oscillations with different frequencies. So you have the very slow ones, the infra-slow ones that are way below 1 Hertz. Then you have the delta oscillations, which is 1/4 Hertz, theta. Then you have the alpha and mu rhythm, beta, gamma, the fast, and so on. And these classes of oscillations have been coined empirically. There was no objective reason to say alpha is between 8 and 12. It was simply observation of brain activity and manual classification, as it's often done in biology. Now, what you have to know is that the interesting phenomenon is that there is a grading of the speed of these oscillations as a function of our cortical state. When you sleep, you engage in the very slow oscillations here on the lower side. As you start to wake up, you start to move to higher frequency oscillations. And when you engage in very intensive cognitive processes and perception, you engage in the fast oscillation. So just like a computer clock, the frequency of the oscillations is somehow associated to the processing needs of the brain, to the processing power it requires. Another important issue we need to discuss is what happens to multiple oscillations. So if you have, these are called coupled oscillators. So you have two oscillators which can be coupled in different ways. They can be coupled in phase, where they go together and there's no phase difference between the two. They can be coupled in antiphase. When one is up, the other one is down. They can be phase shifted, such that they have the same oscillation frequency, but the phase is a bit different. So there's a constant delay. And there is one case, and this is the most interesting one, when you don't have coupling between them. When the ratio of the two frequencies is an irrational number. And there is a reason why this counts and why this matters. If the ratio of the two frequencies is a rational number, then you get what is called generalized phase locking, M-to-N coupling. When one oscillator goes M cycles, the other one goes N cycles, and they meet. If the ratio is irrational, then they cannot meet periodically, and they cannot synchronize. So people have studied this mathematically. There's a very good book by Rosenblum and Pikowski And they studied under what conditions different oscillators can be coupled, or what is the energy necessary to couple different oscillators. So these are called Arnold tongues. What you see here is the energy required to couple oscillators, and this is the ratio of the frequencies. You can see when you have rational numbers, the two frequencies, then the energy necessary to couple the two oscillators becomes minimal. So if you have 2 to 1, 1 to 1, 2 to 3, 1 to 2, this is called end-to-end coupling. As soon as I depart from this regime, it is almost impossible to couple oscillators. You need a huge energy. You need one oscillator to enslave the other. And this is not the case in the brain. The brain is not a strongly coupled set of oscillators, right? Okay, so this leads us to the discovery of why these frequency bands that have been observed for 70 years exist. And some people had the idea to put the central frequency of these bands on a logarithmic scale. And what they discovered is that the ratio between every two consecutive frequency bands that have been empirically defined is the irrational number E, Euler's number, which means that two classes of oscillations cannot couple because their ratio of frequencies is an irrational number. The energy required to couple these oscillators is very, very high. This is why they coexist. This is why the first plot that I showed you, where you saw an alpha wave and the gamma wave in the monkey brain, this is why the two can coexist and they don't interfere with each other, because they are related by irrational numbers between the central frequencies of the bands. So things that for 70 years people have observed now have very precise explanations in terms of mathematics and physics. Now, what are the properties of brain oscillations? What you see on the screen are oscillations that were recorded in my brain with EEG in our lab. And the immediate interesting observation is that this thing, which is an oscillation, it's called an oscillation burst or a packet, it's finite in time. The brain doesn't evolve according to sinusoidal waves that are infinite. It produces packages of oscillations which are localized in time. This is very important. They are time limited. And this is yet another trace that is zoomed in. And this is again my brain. So you see an alpha burst here. Then you see a much faster, higher frequency burst here that is zoomed in here in gamma. And then you see a beta burst that is intermediate. What you immediately see is that the duration of an oscillation is related to its frequency. The lower its frequency, the larger the duration. The higher the frequency, the smaller the duration. It is as if I would have the same process at different frequencies, it's just scaled in time. And this is important because we believe that oscillations behave as fractals in the brain. That it's one and the same process that is simply scaled across different time scales. And this is something we are now actively exploring, and we are now developing tools to detect these oscillation packets such that we can characterize them. Another very important property of brain oscillations, and this has been observed by Walter Freeman, is that if you look at the power spectrum of the oscillations, and you plot this on a log frequency, log power plot, it seems to be a straight line. This is called the 1/f spectrum. Such spectra are characteristic for self-organized critical systems. Systems that are close to criticality. They're not stable, they're not chaotic, they're someone in between, somehow. Importantly, in nature, there are many systems that exhibit power laws. Earthquakes, frequency of accidents, avalanches on the mountains exhibit power laws. What is even more interesting is that if you characterize these power laws with this formula, and you get this exponent alpha, this exponent seems to be almost universal in nature. It's 1.5. Why exactly, we don't know. It's another mystery of nature, but apparently the brain is also part of this organizing principle, universal organizing principle. Okay, so now that we have defined oscillations, we have talked about frequency bands, about the properties of oscillations. The question is, how do we see them? And I would like to strongly argue for the fact that we know so little about oscillations because we were not able to measure them properly until very recently. And as you will see, measuring them is very difficult. So what you typically do when you try to quantify an oscillation is to compute what is called a time frequency representation. So you have a signal, and this signal has this picture associated to it, which characterizes it. So there is time on the x-axis, and there is frequency on the y-axis, and there is power or strength of the oscillation in color code. So blue means it's weak, and red means it's strong, right? So looking at this time frequency plot, you can see that here around about 30 Hertz, there's a very strong burst of oscillation at about 100 something milliseconds. This particular plot is actually showing what happens in the brain of a subject who is looking at a visual stimulus that suddenly appears on the screen at moment 0. What happens is that the signal takes about 100 milliseconds to travel to occipital cortex, and this is what you see when the occipital cortex actually reacts to the presentation of the stimulus on the screen. And you see it has a characteristic frequency, not 1 but 2. It has this 30 Hertz burst, and then it has this alpha burst. And then somehow the alpha is suppressed later on, right? So you can interpret what happens with the oscillations, but looking and computing such time frequency representations. They are the most widely used ones. Now, how do you compute such a representation? There are many techniques. The classical one, the easiest one, is to take a window, a sliding window, and move it through the signal. And for each window, you compute a Fourier, discrete Fourier representation, and then you plot this on the y-axis, and then you have the frequency, and then you have time. The problem with the Fourier transform is that it's using a window that is fixed. You choose it. You choose, for example, 200 milliseconds. What you see here is that apparently you have two different bursts at two different locations in time, but you cannot resolve them in frequency with a short window, right? Whereas you can resolve these two guys in time well, but if you increase the window, you will suddenly see that there are actually two bursts here at two different frequencies, and you lose the ability to characterize the other two that you have been able to discern in time. Which means computing a Fourier representation always involves a compromise. And you don't know where to make this compromise. It's a choice of parameters. So what you see is what you want to measure, what you want to see. You make a choice of a window, and then you're stuck with it. Another option that people frequently use is to use wavelets. Wavelets are packets of oscillations that are simply scaled. This is why they're actually fractals, right? It's the same process that is replicated across all the scales. They are self-similar across scales, and what you do is you take this burst and you slide it through your signal such that the wavelet searches for itself in the signal. And what you get is so-called scalograms. What you see about this is that it becomes narrower and narrower in time as you go to higher frequencies because the wavelets are shorter, but it becomes worse and worse in frequency. So if here things are fine and I can discern frequencies, here it's elongated along the y-axis. I cannot discern frequencies. I can look in time, but not in frequency. So again, I have a trade-off. I have here good frequency resolution and good relative temporal resolution. Here I have good temporal resolution, but very poor frequency resolution. So again, I'm doomed. Again, I have a choice of how I choose the parameters of these wavelets, and I cannot really see the oscillations properly, right? And this happens because there is actually a physical law that governs, much like in quantum physics with the speed and the position of the particles. We have exactly the same problem with the localization of a signal in time and frequency. If you have a short window, you can localize well in time, but poorly in frequency. If you have a long window, you can localize well in frequency, but poorly in time. This is called the Highlander-Gabor limit, and it states that with a single measurement, you cannot localize a signal well both in frequency and time. So there's some kind of curse. So then the idea came, and actually I dreamt this one night, this is why I put this picture, to combine multiple representations. How about using long windows for having good frequency resolution and short windows for having good temporal resolution, and then combining them somehow? Actually, many of the things that we discover in science have been discovered before. Actually, the same idea is applied in optical super resolution technique for which people got the Nobel Prize a few years ago. So this idea is not new. The way we applied it is new, but the idea of combining multiple measurements is not new, right? So how do you combine different representations? So you have a long window, a short window, and then what you want is their intersection. So intuitively, you can compute the geometric mean, and this is how we did it initially, intuitively. Then we later discovered that people have proven this mathematically 20 years ago, and they used it for different kinds of representations. Anyway, the idea is to use the geometric mean, so intuitively to correlate these windows, so you get precision in both time and frequency. And this is how we started the long journey of developing time-frequency super resolution. And these are the people to blame: Adriana de Bucan, Harald Burzan, Vlad Moca, with whom we actually worked on the initial definition of the superlets, which are the time-frequency super resolution. So just to give you an idea of how this works, it's simple. You take multiple wavelets. So at a certain frequency, you can take a short wavelet with a few cycles, an intermediate wavelet with 10 cycles, or a longer wavelet with 20 cycles. And what you see in the Fourier spectrum is that the wavelet that is the longest has the narrowest frequency fingerprint, the black one, right? Here. And the wavelet that is the shortest has the widest frequency, so the poorest localization frequency. So again, you see good temporal resolution, poor frequency resolution, and the other way around. So, and the idea is to compute a set of wavelets with different number of cycles, such that you combine the good temporal resolution with the good frequency resolution, and to combine it geometrically. And this is called the superlet. It's a set of wavelets with a number of cycles that increases in a multiplicative way. I will not go into the mathematical details, but intuitively what you do is you simply compute a geometric mean of these different representations. And what you get is the following. If you use a single wavelet, you have this kind of representation. So you see you have a good temporal resolution, but poor frequency resolution. It's localized in time, it's not localized in frequency. On the same data, the superlet can actually show you that you have multiple different frequencies, and at the same time, it does not lose the temporal resolution. So this is still not too wide and well separated in frequency. And you keep the temporal resolution of the wavelet, but you increase its frequency resolution, right? Here's another example. This is EEG data that I've shown you. So look at the classical representation on the left that people have been using for 50 years, and look what the superlet is showing you. Things that you could not see before. It's like opening your eyes for the first time. You see such a richness in different localized bursting thing, especially in the high frequencies. You see multiple components that are really not evident in the original transformation. Then this is another example on synthetic signals. So you have here signals that are neighboring in frequency and neighboring in time, and they are localized at different moments in time. And what you see is that with the Fourier transform, you either have good temporal resolution, some good frequency resolution, but nothing in between. With the wavelet the same, but with the superlet you always get it right. You can always identify these oscillation bursts, right? Here is another example, EEG in human. And this is the Fourier transform, this is the wavelet transform, this is the superlet again. You see that no matter how you tune the parameters of the classical techniques, you can still not reveal the details that you have from one snapshot with the superlets. This is data from mouse visual cortex. And what you typically do, you present a drifting grating on the screen, and then the brain engages into gamma bursting that is matches the passing of the bars through the receptive field of the cells. You see that with the superlet representation, you can get the best out of it. And not only, but if you zoom in, you can see lots of components in your spectrum that identify components that you totally miss with the classical techniques. You're completely blind to these details with classical tools. Okay, so let's list a few of the properties of this technique. One of the most important properties is so-called reduced spectral leakage. So what you see here is that you are computing the representation of the same signal with three different methods. This is the Fourier transform, this is the wavelet transform, and this is the superlet. And you see that in both cases you have leakage of power from the frequencies below or from the frequencies above, but not with the superlet, because the superlet concentrates time frequency estimation so well, it doesn't suffer from the spectral leakage that actually blinds you. And if you look at these details, you cannot recover them from the classical techniques. Right? The second property is called non-dilution. One of the big problems of analyzing EEG data in humans is that the higher you go in frequency, the weaker the signals. The reason for that is that the bone and the skull acts as a low-pass filter. So the fast oscillations in your brain, even if they're present, you cannot measure them. Or you can measure them, but they're really weak. So they're strongly attenuated. On top of this, what happens is that the classical techniques also dilute your representation. This is the Fourier-based representation. You see that you have these three pieces of your representation, and you see that as you extend this representation more and more, your burst here will be more and more diluted out. You don't see it anymore. So not only your higher frequencies, your gamma that is associated to cognitive processes and that you want to study in psychophysical experiments, not only it's weak and diluted by your skull, your technique is also suppressing it even more, so it makes you blind to it completely. This is why people doing EEG have frequently said there's no gamma in human EEG. That's entirely wrong. There is gamma in human EEG, and not only there is gamma, but it's beautiful and well and strongly expressed. So the superlet doesn't suffer from this dilution property. It can reveal your oscillation no matter what the range of exploration is. Third, it has what is called a powerful time frequency concentration. So we did an experiment. This is EEG from humans watching some dot stimuli on the screen. And we took 84 trials, 84 signals, and we inserted in only one of them packets with a certain time frequency structure that is known, so a kind of ground truth. And then we computed different representations and tried to recover what we inserted. And you see that these other techniques, the Fourier technique, this is a short-time Fourier transform, this is a super-resolution Fourier technique, this is the wavelet transform, sometimes can recover some of the components, but not all of them. The only method that can reliably recover what you put in the signal is the superlet. So it can recover your signals even if they're hidden in one out of 84 signals, right? Property 4 is that they have unsurpassed detection capabilities. So this is an experiment that we did with real data, where again you insert the target and then you want to see your capability of extracting it if you increase your noise. So you have a signal to noise ratio that decreases. So the more cluttered the representation is, you want to see how you are able to recover what you put in. And again, the representation that has the best detection capability is the superlet. Finally, this is one very interesting property that somehow is related also probably to quantum physics. The superlet representation is able to concentrate frequency representation based on context. So if I have a single packet, the superlet representation looks like this. If I have two neighboring packets, then the neighboring packet helps me concentrate, gives me evidence about the likely representation in frequency of the other packet. So you see there is contextual inference, because if you have two packets that are neighboring at the same frequency, that means you most likely have a single signal that was interrupted, which allows you to even better estimate its frequency, right? So this is a property that I think no known time frequency representation has, at least not in my knowledge. Finally, in order to validate that this technique works, we had to perform complicated experiments in mice, and this can be done using optogenetics. So there is one technique where you actually make the cells express channels in the membrane that are sensitive to light. And then you can shine a laser with a different frequency and induce a certain oscillation. So here you see light stimulation, and here you see the time frequency representation of the signal that you produce. And you see that the other techniques are virtually blind, but the superlet is revealing beautiful oscillation packets. And then we validated that these oscillation packets are truly oscillatory bursts. Not only that, but we discovered something very interesting that was not known before. That when you shine light on and off, you basically engage the cortex twice. You engage it when you shine it on, and you also engage it when you shine it off. So if I stimulate with 100 Hertz, I get a response at 200 Hertz. The problem is, people don't know or don't believe that oscillations with such high frequencies exist, so they never look there. But what we discovered is that you can reliably entrain 200 Hertz oscillations, which are very rarely seen. Okay, further details you can find in the paper. What is nice about this technique, when we invented it, it was simply a tool for neuroscience, for seeing oscillations in brain signals, but people have started to use it in other fields of science. People are now using it to design diesel engines, to study the shaking of skyscrapers, to study oscillations in the magnetosphere, to study, for example, in India, the failure of power grids. So when a power grid is failing, it exhibits a transient oscillation that is predictive of the fact that your power line will go down. And apparently the superlet is good at picking this up. And so this is something very nice for a scientist to see that what you invented for a certain field is then picked up and applied in a wide range of other scientific fields. Okay, so the last part is related to why we talked so much about oscillations. And the strongest indication that these oscillations are actually useful for the brain is the fact that when you have brain disorders, oscillations are also disrupted. It's a kind of a causal marker, if you want, or biomarker of the fact that oscillations are involved and supporting normal brain activity, because when the brain activity is disrupted and not normal again, oscillations are also not normal. So what is known, for example, in schizophrenia, that if you measure a healthy brain, then you get oscillations in different regions of the brain, and they are synchronized. You can do experiments in healthy humans. If you do the same in schizophrenic people, you can produce localized oscillations, but they don't synchronize anymore. So this led to the idea that oscillations are related to information transfer and coordination of brain assemblies. What is also interesting is that predominantly the fast oscillations are disrupted. And now there's tons of evidence that gamma oscillations and beta oscillations are disrupted in schizophrenic patients. So then we started to study this in our lab, because we study oscillations. It was interesting to actually relate these to schizophrenia. There's one hypothesis of a type of schizophrenia that appears, and that is that excitatory transmission in the brain is disrupted. For example, there is one receptor, which is called the NMDA receptor, that may not function well in schizophrenic patients. So what we did was to bring some mice from Heidelberg that exhibited a knockout of a subunit of a receptor of this NMDA receptor. So such that their excitatory transmission in the brain is impaired or less efficient, right? First, we tested if these schizophrenic mice are different from the normal mice. And Anna did experiments with measuring how much immobility a certain mouse exhibits when you put it in a state where it needs to run away. What's interesting is that schizophrenic mice are much more immobile, freezing, than normal mice. Normal mice just try to run away and that's it, to escape. Schizophrenic mice become catatonic. They simply freeze and they try for a while to cut loose, but then they stop. So we proved that there is a behavioral difference between the two cohorts of mice. The next experiment was to try to see what happens to the oscillations in the gamma oscillations in the brains of schizophrenic mice versus healthy mice. And one idea is that you could try to recover the brain oscillations by actually enhancing this receptor that you know it's destroyed. It has, the NMDA receptor has all these subunits. The one that is impaired in these mice is the GluN2, the green one. So what you can do is to try to compensate. You give it a substance, in this case it's called diserine, which is binding on the other side that is intact, and it's recovering the function of this receptor. So if it's broken, you ramp it up again and you compensate on the other, using the other subunit. So this is exactly what we did. We performed experiments in mice that were receiving visual stimuli. We measured how they react to visual stimulation with a blind or whatever neutral condition without applying the substance, and then we applied the substance directly into the brain. What you see is that these gamma oscillations that are expressed become much, much stronger, which means using this, it's a supplement that people take, especially in US, people who do sports, this D-serine, is able to strongly recover the gamma oscillations that you know are disrupted in schizophrenic patients. And this was actually inspired by the clinics of psychiatry in Jerusalem, who were doing experiments with D-serine with humans with schizophrenia. And they found, at least in one patient, that administering this substance actually completely alleviated all the markers, all the measurements, and they were indistinguishable from the ones from a healthy subject. And this actually led us to try to explore what's going on. More, we did experiments in schizophrenic mice and healthy mice. And you see that in healthy mice, D-serine doesn't play much role. So in schizophrenic mice, D-serine really restored the gamma oscillations that are disrupted in schizophrenic patients. Now, this is only at the beginning. We are still working on this project and trying to do more experiments and to try to come with an explanation why this happened and try to prove that actually you can recover not only the brain oscillations, but also the normal behavior of the mouse. Another disease that also exhibits a disruption of oscillations is in Alzheimer's disease. What happens in Alzheimer's disease is that you have some proteins that accumulate in the brain and actually destroy your neural circuits, make the neurons die. Unfortunately, about 10% of the population over 65 has Alzheimer's, and about 30% over 85. Only in 2019, we had about 70 million people affected, and this number is going to strongly increase in the future. Okay, so not only oscillations are disrupted in such disorders, but people from MIT have discovered that if you apply a flicker, visual flicker, to mice that exhibit Alzheimer's disease, suddenly the brain clears up, and in about a few weeks of treatment, they recover normal function. Something that is so trivial. All of you know the stroboscope, or this kind of flickering light that people use for special visual effects. Apparently, if you use a frequency of about 40 Hertz, you can restore normal function in early incipient Alzheimer's disease in animal models. Now, this is called the genus therapy, or gamma entrainment using sensory stimulation. So what you do is, this time, it's not an oscillation that the brain produces, it's an oscillation that you impose on the brain. And apparently this helps these amyloid plaques clear up. What happens is most likely that you engage in gamma oscillations which are metabolically intense, and they activate the glial cells, which then start to initiate a cascade, a metabolic cascade that clears up the brain. Moreover, people have discovered just a few months ago that this is efficient not only in Alzheimer's, but also in dementia. Also, if you use flicker, it can increase your cognitive capabilities. Why this happens, we don't fully understand. But we started to experiment with this in our lab. And we have a project together with the University of Oslo that is funded by Norway grants, where we initially optimize the therapy in mice, and then we transfer it to humans. And the idea is the following. Not every brain has the same resonance frequencies. You remember we talked about resonances in the beginning. The brain reacts to external stimuli better if it resonates to those stimuli. And if a brain resonates at 40 Hertz, another brain resonates at 50 Hertz, another brain at 60 Hertz, and we are all different. So what you need to do in order to optimize the therapy is to optimize it for a certain individual. And we discovered in mice that we have the same problem, that different mice have different resonance frequencies. But we also discovered something very important, that the color of the light is essential. And in this case, this was an interesting and unexpected discovery. If you use blue light in mice, you can induce gamma oscillations that are significantly stronger than if you use white light. In humans, probably red light works better. But what is interesting is that the color of the light influences mostly the higher frequencies. This is again something that is very incipient in our lab, and we are still working to explore it and to fully explain it in the next months. But the take-home message is that you can use oscillations and entrain the brain in oscillations such that you can treat diseases, right? And this brings us to the conclusion of my talk. So the take-home message is that brain oscillations are very complex processes. They emerge from very complex mechanisms. They're not easy to oscillate, to estimate. And in particular, because they occur in such tiny bursts, you need specialized techniques that can actually discover them and extract them. And again, the message is that we have so far been blind to many of these transient oscillations because we did not have the tools to properly measure them. Then what we know is that the disruption of oscillations can be a hallmark of disease. So people are now thinking about this, devising biomarkers that are predicted. So for example, the disruption of a certain oscillation band could predict that in 10 years you may become schizophrenic. And then you can do early intervention to prevent the onset of a certain disease, right? Finally, oscillations are not only endogenous to the brain, but they can also be used to treat disorders. And this is something very surprising. And it's really catching a lot of ground now in different labs in the world to try to use oscillations to heal the brain. Okay, thank you very much for your attention, and I'm looking forward to the questions.
Thank you very much for this really fascinating talk. A lot of new information in a short time, but I think we learned a lot. But let's move to discussion session. I am telling the online audience that there are present here a few collaborators of Raul and other colleagues interested in this topic. They also followed the presentation, and maybe they have some comments or questions related to this topic. We have here a microphone, so if you have questions, comments, please use it in order to the online audience who can hear the question as well.
Sorry, very nice talk. Few questions which I'm working. I don't know if you are saying the oscillation, brain oscillation. In fact, you consider that are electric oscillations, not other kind of oscillation of the cells, that mechanical. Yes, so it's a matter of electric oscillation. In this case, you consider that it's good that there is some relation between the frequency of stimulators and the brain, particularly the brain. But what about the frequencies particularly so in this electric oscillation of the brain? There is such a correspondence between the excitations and the behavior of the brain electrified.
Actually, we know a lot about the relationship between the frequency of the oscillation and particular brain states. For example, we know that during sleep, when we go to sleep, we start from a normal aroused state where the brain mostly exhibits oscillations that are higher than alpha, beta, gamma, so about 30 to 80 hertz, if you want. The gamma range is traditionally defined between 30 and 80. Mostly in the awake state, in the cognitive aroused state, like we are now, we are in this state of higher frequencies. When we start to go to sleep, what happens to the brain oscillations is that they start to slow down. So initially we become drowsy, we go into alpha, then the delta oscillation emerges and becomes more prominent. And finally, when we go to sleep, we dive down into the slower oscillations. Now, the sleep that is mostly useful for us to get a good rest are the deep, slow oscillations. Paradoxically, there is a state of sleep where the brain kind of wakes up. It's called REM sleep, or rapid eye movement sleep, where the brain's oscillations are suddenly becoming very fast again, but you are still sleeping. And it's called paradoxical sleep, because although you are sleeping, you are actually dreaming, and your brain is kind of awake, but it's locked in. During this paradoxical sleep, it's also very dangerous for the body, because you're virtually paralyzed. It's the state in which it is the hardest to wake up from external stimuli, right? So there is a very strong correlation between what happens to us in terms of perception and behavior, and the frequency of these oscillations. Recently, people have discovered that there is a relationship between the delta oscillation, which is the initiation of sleep, and the frequency that a baby feels in the mother's womb when the mother is walking. So and you know that you can put the baby in a cradle, and how do you call this? You can put it in a swing, and if you put the baby in a swing, even a human, it goes to sleep. And the reason is that the mechanical oscillations that you produce are then translated through the vestibular system and other systems to the brain, and entrain the brain in delta, which then initiates sleep and brings you to the slower frequencies, right? Then, with the higher frequencies, we know, for example, that higher frequencies are expressed not only in the awake state, but also under meditation. And there are experiments with Tibetan monks who are actually engaging into strong meditative states. And if you measure with EEG their gamma power, the high frequency oscillations, there are orders of magnitude above that of a healthy human, normal human that is not trained. Not only it is helpful for this, but apparently gamma oscillations are useful also for enhancing your cognitive abilities. There's one experiment of the termite. So you have a group of ants or termites, and then a normal human that is untrained can follow one or two, or a group, but cannot follow 50 termites at the same time. Through meditation and training, these monks actually develop higher cognitive abilities, and they can, this is a measured thing, it's a known thing, it's not pseudoscience. This was published in PNAS, in the Journal of the American Academy of Sciences. So when they engage into these strong meditative states, their gamma oscillations are also orders of magnitude higher. So now we know that there's a relationship between your cognitive load, your behavior, and your arousal or sleep state, and the frequencies of these oscillations. So there's a clear correspondence. So this leads us to believe, at least the people who study oscillations, that they really count in the brain. They're not an epiphenomenon, there's something that supports brain function.
Thank you. We also have a question from the online space. I will read it. Actually, the question has two parts. One, is there any relationship between different types of brain oscillations and shapes of action potentials of various types of neurons in the cortex? And two, are there spinal cord oscillations?
Yes, thank you. So related to the first question, that's a very good question. Actually, I don't know if anybody studied the relationship between the shapes of spikes and oscillations. What we know, for example, is that different cell types are engaged in different kinds of oscillations. The fast oscillations are produced mostly by the fast spiking basket cells that have narrow spikes that last 0.8 milliseconds, and they can fire in fast succession. The slower oscillations seem to be more produced by this kind of network bursting mechanism that is likely produced by the pyramidal cells that have large action potentials. Now, if a cell can change the shape of its action potential as a function of the oscillation of the circuit, I think nobody has looked at this. So it would be very, very interesting to study. And also because there is still the dogma that individual cells fire stereotypical action potentials. We know from our experiments that this is not the case, that the shape of the action potential changes in time. Now, how this could happen with different oscillations, it's something very, very interesting to study. And I think it's a very good research question for the future. Regarding the second question with the spinal oscillations, yes. For example, it is known that you can measure coherence between motor cortex and the muscle spindle that you get on the muscular potential. Interestingly, when you reach and when you move your arm, the highest coherence between motor cortex and the muscle potential is in the gamma band. This leads us to believe that the gamma oscillations are somehow multiplicated at all the levels, from the brain to the spinal cord and to the muscle. So there are measurements that indicate clearly that this is the case.
Thank you. If there are other questions. If not, I have one more question from the perspective of the new neuroscientist generation. Last weekend we have some high school students visiting our faculty, and one of them told me that he wants to be electrician, brain electrician. And he told me that he loves biology, he loves informatics and physics, and his question was, what is the best way to achieve his dream? So maybe you can give some advice for him. Maybe he's looking to us. And
well, my main advice is that you can learn to study the brain no matter what your background is. So I studied computer science, and then I started after a few years to do experiments on the brain, to do surgeries, to implant electrodes. I did not have the proper background coming from college. I did not have the training to do it. But if you're really passionate about it and curious, you can learn anything, and then you can get specialized to do it. So my only advice for him is to not give up, to actually go to a place where people do such experiments, to go to an interdisciplinary research institute, like the Max Planck is, for example, in Germany. In UK, you can go to UCL and so on. And go and learn the techniques, and do this early. Don't wait, and don't be shy about your background. I think it's something that anyone can do, and it's fascinating.
Thank you. Thank you once again for the presentation and for discussion part. And we also thank you, the members of the Babeş-Bolyai University community, for being with us. And we are looking forward to meet you at the future presentations within the Studium Generale program of the Star UBB Institute, which is transmitted live from here, from the Taylor's Bastion of Cluj. Thank you.